Okay, so this is my video on reviewing a scientific journal article over my protein pepsin. The first scientific journal article I found that I'm potentially going to use in my paper is called A History of Pepsin and Related Enzymes. The author is Joseph F. Fruton. He studied at Yale University, and the part that I looked at was volume number 77, number 2, that was written in June of 2002. Now the abstract is on here, but I looked at the full article on a different computer. This is from the book The Quarterly Review of Biology. And I found I'm going to include the link on my submission, by the way. What I found in this article so far that I was going to include in my paper was a lot of history, and that's stated in the name, A History of Pepsin and Related Enzymes, but it also had some other information, including how they found the enzyme and how they found the structure. This was more foundational, such as finding out different ways of being able to observe small molecules in proteins. Pepsin was found when there were studies being conducted on gastric digestion during the between 1820 and 1840. It is an agent and where the pressure of stomach acid causes the dilution of nutrients, such as meats or coagulated egg white. That's similar to what I already knew about pepsin. What I knew already was that pepsin is the activated version of pepsinogen, and it's produced by the proteolytic enzymes. Those are produced by the body chief cells, and those are found in the submucosal tissue layer of the stomach, in the pyloric and the fundic regions. Now, I'm going to name a couple of people. I'm not sure, I probably will in my paper. But 1 year after Johannes Müller published a paper on pepsin, Theodor Schwann published a paper stating that the fermentation of sugar by yeast is affected by a living microorganism. And that was based off of Müller saying it was possible that digestion is different from yeast fermentation, but a ferment was still involved. This was before they actually knew about pepsin. And pepsin, they found out how to isolate pepsin by using lead acetate to precipitate pepsin from an aqueous extract of carefully washed gastric mucosa. The lead was removed by hydrogen sulfide, adding alcohol to the remaining solution, and then drying the precipitate. Here's a little picture about digestion, just what I knew. Here's a ribbon model of pepsin. Now the 3D structures of pepsin and others related to it were found through diffraction techniques, and this was helped in seeing the mechanism of the catalytic action of these enzymes. So when they were able to find out what pepsin looked like in a 3D structure like this, and other structures like it, they were also able to observe the catalytic action of the enzymes. The amino acid sequences of many proteins, including pepsin and pepsinogen, were determined when new separation methods were introduced. And those separation methods were most notably chromatography and electrophoresis. Now, learning about this has helped to lead to the design of new inhibitors of aspartyl-proteinases, which are participants in the maturation of human immunodeficiency virus and in the generation of Alzheimer's disease. So it looks like I'm going to have a couple of diseases in my in my paper. Thank you.